talk about next are different applications of GemPros that have been used and developed within the last 10 years in our lab. And so the first one that I think is pretty easy and simple to understand is using the structures of proteins to understand what their function is catalytically. And we had this question during a metabolic reconstruction of a cyanobacterium recently. And the question that came up here was that there were 4 predicted enzymes that were predicted to carry out the same function. And they were all considered to be essential in gene essentiality experiments. So the question was, which one really is carrying out this one reaction in our metabolic network? And that reaction was a phosphoglycerase mutase. And we were able to construct homology models of these 4 enzymes to try to understand if there are certain residues that allowed functionality for this specific activity, which was a phosphoglycerate mutase. And so we were able to dive into the literature once we generated these models, and we found that there are specific residues at specific positions that conferred this functionality. And with these models, that allowed us to place the residues in 3D space of these different enzymes, which were different in terms of their sequence identity. So a sequence alignment wouldn't tell us where the position in 3D space of this certain residue was. And by doing that, we were able to separate the function of these all similarly annotated enzymes into specific functions defined by the locations in space of these residues. And so that allowed us to more confidently place these enzymes within a metabolic network in terms of their catalytic activity. So the next application, like I mentioned, was the first application of a genome scale model with protein structures. So in this study, they reconstructed the metabolic network of Thermatoga, and the evolutionary question they wanted to answer here was, how do proteins evolve within a metabolic network? And the way they approached this was to look at the folds. And so if you remember from the last lecture, the folds are kind of just the basic shape of a protein and how they fold together. With these folds defined for every single enzyme within a metabolic network, they wanted to understand, do metabolic networks evolve by recruiting their neighbors? So their neighbors may look the same as them and maybe carry out a similar function, so the next step in a metabolic pathway. Or do they recruit kind of these promiscuous other enzymes from other parts of your metabolic network to help you carry out the next steps of your metabolic pathway? And so these were called the retrograde or patchwork models, where the retrograde is your neighbors and the patchwork is recruitment of enzymes further away from yourself. And they found that just by looking at the protein folds, that the folds were actually very different within within your neighboring enzymes, which kind of disproved the retrograde theory and proved that more of the patchwork theory was applicable in terms of evolution of a metabolic network. And so this was all done with structural genomics and putting structures in the context of a metabolic network, which could not have been done with just either one of those. So if you just had the structures, you couldn't really understand where their position was within a metabolic network and understand how that was evolved. And with just a metabolic network, you couldn't understand the protein folds because you didn't have the structures to get that information. So this was really a unique application, I think, that kind of jump-started GemPros as a way to answer simple evolutionary questions like these, and then use them as replacements for the edges in your network to get information from the structural properties to help simulate with them. So when I joined the lab, the first thing that we did was we tried to replicate these GemPro pipelines and apply them again to Thermatoga and also a previous application of E. coli. And what we applied it for was to understand the differences between the proteomes of these two organisms. So obviously Thermatoga maritima lives at a much higher temperature than E. coli does. And we wanted to first calculate a number of structural properties for all the proteins within these metabolic networks and compare them based on their positions and the similar reactions that they were carrying out. And so the properties that we calculated were, again, simple ones like we mentioned in the last lecture, like where the charged residues are, how many surface residues there are, the basic secondary structure of enzymes and things like that. And so what we did was a simple PCA analysis of these properties based on their positions in these metabolic networks of E. coli and Thermatoga. We were able to split them up based on these properties and their reactions that they carried out. And for instance, this example popped out to us, which is I can't see the word. I think it's PGK, phosphoglycerate. Well, not phosphoglycerate, it's PGK. It's an enzyme in E. coli and Thermatoga. But what we saw was the properties of these two enzymes, which carried out the same reaction, were very different because Thermatoga required it to be much more stable at a higher temperature. And that popped out to us in this PCA analysis because of the structural properties that characterize these two enzymes. Yeah?
Can you just say what's PCA analysis?
So principal component analysis was used to kind of take a very high dimensional set of data. So we had many different features of these different proteomes, right? And then just break it down into kind of the reduce the dimensionality of that data set to try to understand, first of all, to visualize it in 2D space, and then to try to see what properties were contributing the most to these different components. So that was used to take kind of a larger, harder to analyze data set and break it down into simpler parts. And so another application of this same method was used to classify 55 E. coli strains, which were, I think you guys have looked at this study for this class. And we tried to classify these 55 strains based on their structural properties as well. And what we found was we could actually classify them based on the simple charges of their amino acids on the surface of their proteins. And that allowed us to easily classify these strains of E. coli based on their virulence properties. And with the structures, that also allows you to kind of dive deep into what these properties may be influencing in terms of the structure itself. So that allows you to look at specific mutations that may be causal for different virulence properties and start investigating those kinds of things. All right, so next up, I'm going to talk about the work of Roger Chang, who was a previous PhD student in the lab, whose main work was also GemProbes. So what he looked at were kind of, I consider this in two directions. One is a retrospective direction, and one is a more prospective direction. And so what he first looked at in terms of the retrospective direction was he wanted to understand why a clinical trial had failed for a drug. So this was a drug that had gone through, I think, 2 or 3 phases of clinical trials, and then it had failed at phase 2 or 3, costing a pharmaceutical company $1 billion or so. And obviously, if you're able to avoid that, that would be a good thing. And so with the GemPro, what Roger did was he took the structures of human enzymes and he predicted their binding sites for this certain drug based on these structures in using a geometric method. And what he did was he took these predicted targets and inhibited them based on their position within a metabolic network. And so with these simulations, he was able to recapitulate the kind of side effect that was seen in the clinical trials, which was, I think, high blood pressure, which was fatal for this certain disease that the drug was treating. And so a GemPro allowed him to not only predict what other proteins this drug might bind to, but predict their phenotypic effects if they had bound to what these are called off targets. So these are the targets that your drug, you don't know what these targets are that this drug may bind to that may cause fatal side effects. And so a GemPro was applied in a retrospective fashion here to understand what the true effects of a drug were. So prospectively, what he also did was he took in terms of antibacterials. He wanted to understand if we could target new proteins with different antibacterials. So in the opposite direction, he took a genome scale model of E. coli, simulated it, did some gene deletions to simulate inhibitions within the network, and from that, took these targets that could be inhibited to cause death and antibacterial effects within the cell. Took the structures of those enzymes, ran the same kind of similarity analysis for drugs to bind to it, and then came up with candidate antibacterials based on this workflow. So these are two, I think, really interesting applications of a GenPro because, again, you can't do this kind of stuff if you just have the structure or if you just have the metabolic network. You kind of need a synergy between the two to understand if an effect is going to happen based on things like pharmacology interactions and stuff like that. So these really elucidated what is called the mechanism of action of a drug, because what you can do is true multi-scale modeling of side effects or an antibacterial. So when I came to this lab, another application I was interested in was trying to apply more detailed methods like molecular dynamics and docking, like we mentioned in the last lecture, to understand the effects of what a mutation is really causing on a system and a structure. So what we did was, in this study, we took a couple of enzymes in the red blood cell model, and we simulated their wild type and mutant protein structures. And we wanted to see how that impacted the binding of a drug or a metabolite. And so with MD and docking and free energy calculation techniques, what you can do is you can compute those differences between wild type and a mutant to the binding of these small molecules, and then get a difference free energy of binding for these two enzymes, for the wild type and the mutant, and then simulate that with the genome scale model. And in this case, we actually went all the way to using a kinetic model, because what we get from these simulations are differences in kinetic parameters, like a binding affinity, so a Km in this example. So in this aspect, we really went far into using detailed molecular modeling simulations as well as detailed genome scale modeling techniques. And so we incorporated that information back into the red blood cell to analyze the effect that a mutation had on the whole system. And so I think one thing we learned from this study is that it is definitely, it is possible to apply these kinds of methods on a couple enzymes within a genome scale model, but it is still a couple years off, I think, from applying it on a genome scale for all enzymes, like analyzing maybe hundreds of mutants at once and then integrating that back into a kinetic model. I think is about maybe 10 to 15 years out from now to being truly applicable on a genome scale. So another couple studies that have used MD in a similar way were to try to understand the effect of a post-translational modification on a cell. And so this was done by Elizabeth Brunk, who was a postdoc in the Paulson lab. And what they had was a set of experimental data that kind of showed the phenotypic response that occurred when you had a specific PTM for certain enzymes within your network. And what MD showed here was it allowed Liz to really understand the mechanistic impact that a PTM had on the protein itself. So why would it either inhibit or increase its activity? And then use that to understand if that would be related to the phenotypic response that was being observed. So again, this was applied on a handful of enzymes within the metabolic network. And so yet another study that used molecular dynamics was to try to understand the mechanisms behind mutations in RNA polymerase. So we have a couple of experiments that are called adaptive laboratory evolution experiments going on, where we try to evolve E. coli under different conditions and sequence it and see what has changed within this bacteria. So one enzyme that gets mutated a lot is RNA polymerase because it's involved in a lot of regulatory processes in your cell. And these mutations often occur at different parts of RNA polymerase. So they may be far apart, they may be on one side or the other side. And what MD simulations were able to show were we were able to break down kind of the effect of these mutations into specific chunks. And so we called them neighborhoods of impact neighborhoods, I guess. And what that allowed us to do is kind of classify these mutations in terms of the impact they had on these specific neighborhoods of interactions between the RNA polymerase, and link that back to the phenotypic data that was being shown within these AL experiments. And that was all done with molecular dynamics simulations of this enzyme, either mutated with these mutations. All right, so all these previous studies, I think, considered maybe enzymes either beforehand or at a single scale, at a smaller scale. And so what was done next was to incorporate these properties within simulations of the metabolic network. And one of the first applications here was using M models to simulate E. coli growing under different temperatures. And so that couldn't be done if you just had the metabolic network. You needed to understand how the protein properties differed under different temperatures. So this again was done by Roger Chang, and what he did was he took predictions of the thermostability of different enzymes at different temperatures. He created kind of a Vmax based on this thermostability of these enzymes at different temperatures, and he was able to incorporate that back into a metabolic model of E. coli to enable simulation of the entire model under different temperatures. So this graph here kind of shows you the results of that simulation and how well that matched up to experimental data. And this here was I think, one of the first applications of using protein properties to improve the capabilities of a metabolic model, because if this allowed you to simulate under different temperatures and also explore kind of the growth limiting pathways within a metabolic network based on the temperatures that you were simulating at. So this was quite interesting, and just recently was improved upon in the context of me models. And again, me models incorporate protein folding and other things like complex stoichiometry. So if you can imagine, at a higher temperature, there's a lot of folding that needs to happen for the protein to remain in its correct shape. And so chaperones are the main kind of agent to maintain correct folding within a cell. And so what was done in this model called FoldMe was that the chaperone folding network was incorporated into the me modeling framework, as well as these properties of proteins that are affected at different temperatures. And so what that allows you to do is simulate, again, at different temperatures, but at a much more accurate level now. So here is the line represents the simulation, and the dots are the experimental data. And so incorporating the trade-off between should I remain correctly folded using my chaperones, or should I produce more protein because there's more unfolded proteins being created? This framework allows you to explore that question. And so at a higher temperature, it was found that a cell does produce more chaperones to keep all its proteins correctly folded at higher temperatures. While at a lower temperature, it doesn't really care, and it will just produce more proteins to carry out more functions when it needs to. So this was quite interesting because we were able to now incorporate a lot of different protein properties that may not fully predict our phenotype. So just predicting the folding rate of an enzyme at different temperatures did not allow us to do these detailed simulations, but incorporating all that kind of stuff along with the chaperone folding network allowed you to finally get a computational simulation that matched up to experimental data pretty well. So I think this was quite an interesting study that really incorporated multi-scale modeling in the framework of a me model. Another interesting study that was recently done was to understand the effects of oxidative stress on a cell. And so oxidative stress is when reactive oxygen species like hydrogen peroxide or superoxide are stressing your cell out. And so these are encountered in all forms of life under aerobic metabolism. And what we wanted to do here was we wanted to simulate, again, a me model of E. coli under oxidative stress conditions, so varying concentrations of ROS in our simulation. And one source of damage that ROS will impact are your proteins in your network, as well as your DNA and lipids that make up your membrane. But I'm going to focus on the proteins because that's what this lecture is about. So when you have a protein, it may have a metal binding site, and these metals are often susceptible to being damaged by ROS. And with a protein structure, what you can do is you can calculate the probability of damage based on the location of this metal ion. And so if a metal ion is kind of safely buried away from ROS, it will probably not be damaged. And that was found in a couple studies where a metal binding site was deeply buried and not accessible to solvent within a protein. So what we did was we took that kind of knowledge and we incorporated it based on computations of these structures within the metabolic network and created kind of a probability of damage for these enzymes based on that information. And that was incorporated within the context, again, of the me model to understand if we could simulate a cell under different concentrations of ROS. All right, so those were kind of, I think, the main applications of GemPros within the last couple of years. And these next couple of slides, I'm going to talk about what I think are the main future-facing applications of structural systems biology. And so one of them really is visualization, because what can you do with 3D information? Well, the first thing you want to do is visualize it. And so structural systems biology can help you to answer that question. And so this kind of brings you back to, like in middle school, you would build models of animal cells, styrofoam models and things like that. Structural systems biology can allow you to do that now computationally and accurately. And so there's this method called CellPack, developed by Art Olson at Scripps. And what that answers for you is this basic question of how do I fit all these protein molecules inside my cell? And how do I do that correctly? And so that's a packing problem. So you can imagine an analogy to this is how can I pack all my moving boxes into my U-Haul truck correctly and have them all fit? And that answers, this is a simulation that allows you to do this packing problem in 3D space. And another question that I think would be really interesting would be to apply molecular dynamics simulations to a whole cell. And there are a couple of groups that are doing this kind of work. So here is a simulated, I think, a cube of a cytoplasm of proteins. And they're able to do this for a short time scale. And like I said, I think this may be applicable in maybe 10 or 15 years from now to do this using, say, a packed cell that we have simulated. Another thing that would be super interesting to look at would be to apply this entire framework to human health. And this recently was done in Recon 3D, which is the most up-to-date metabolic reconstruction of human metabolism. And in this reconstruction, we also did incorporate the structures of 3D proteins, and we wanted to understand how do cancer mutations cluster together in 3D space, and can that give us insight into how these mutations are impacting regulation within a cancer cell, and can that possibly give us clues to what we can target with anti-cancer drugs? So this provided us insight that mutations do cluster together in 3D space in certain enzymes in cancer. And that gave us a mechanistic understanding of, say, does this set of mutations impact the interaction between this protein and another to increase transcription or something to create a metastatic cell or something like that. So this was a really interesting, I definitely encourage you guys to check this paper out because there are not just this, there's many more applications than this hotspot study that was done. And gives you really good insight into how you can use structures at many different levels within a metabolic network. And finally, I think another application that we haven't explored, but I think a couple other labs have looked at, is using structures as well as systems to improve metabolic engineering and strain design processes. So obviously, as you can imagine, an enzyme will carry out the reactions to create the chemicals that you're interested in producing in a metabolic engineering process. But you'd also like to optimize your strain to produce that amount of chemical. And so this is where enzyme structure and efficiency and specificity really work hand in hand with the entire network that is creating this chemical. And this is important for a lot of applications, like in green chemistry, where you're producing chemicals that may not be economically eco-friendly to produce without these methods, or you're producing chemicals like artemisinin, which is an anti-malarial drug that only can be found in a Chinese plant and it can only be isolated from there. So producing things like this at a larger scale would be extremely applicable in terms of using a host cell to do so. So I think there's a lot of applications that are being explored by a lot of industrial companies to do this kind of work. And it's really interesting to be able to optimize that stuff based on the knowledge that you can get from protein structures as well as metabolic networks. So finally, I have kind of a table that summarizes all these applications into one in terms of the structural tool used, the systems tool used, and the computational complexity that is required to carry out these simulations, along with the reference link here, so you guys can use this as a reference to understand the applications of GemPros and what was required to do so. So some of these were a little easier than others because they didn't require as much computational time to start the analysis. And some of these, like the MD-related ones, did require a lot of work to begin to set up and also to analyze. But this just shows you, I think, the scale of different analyses that can be done with the integration between structural biology and systems biology. So in conclusion, I just want to leave on the point that GemPros truly enable multi-scale modeling. So they really allow you to inspect molecular interactions at the atomic scale and bring that back up to what's happening on a whole level of a system of metabolism. And as you can see here, that there are a number of wide-ranging applications to look at the impact of mutations to help you reconstruct the metabolic network, to help you simulate and expand the properties of a metabolic simulation and apply them to different conditions. And I think there are a number of unexplored opportunities for the integration of structures in systems biology that maybe you guys can begin to start working on in your graduate career. So with that, thank you for your time. I hope you learned something new about what protein structures are and how they could be used in systems biology. And I'll take any questions.